Hello, I am Dr. Neeraj Rai. I am working on DNA aspect at this institute, and my main focus is to work on human population and animal breeds of the South Asia. So we have two labs. One is ancient DNA lab, and another one is modern DNA lab. So in ancient DNA lab, we are mostly working on archaeological remains of humans and as well as animals. And in modern DNA lab, we are working on modern DNA populations. So our main objective is to reconstruct the population history of ancient India, in which we are looking at the human genome as well as the animal genome level. So we have in our ancient DNA lab, we are isolating ancient DNA and we are preparing the libraries and we are sequencing the DNA. And in modern DNA lab, we are collecting the modern population samples and we are trying to sequence the modern DNA as well to compare the ancient as well as the modern DNA data. The main objective of this work has a lot of application in the domestication as well as the population prehistory of this South Asia. So I'll be talking about the ancient DNA perspective and application of ancient DNA research in this new era of world. Conventionally, we used to use archaeological records, cultural records, anthropological records to look into the past of human beings as well as the animal domestication pattern. But now with coming of the new technology and advancement of the new methods, we can isolate DNA from archaeological materials and from a human remains which has been lived in the past and which represent the population history of our modern day world. So in this talk, so in BSIP we have world-class ancient DNA lab, and we have also collected a lot of archaeological materials from throughout the South Asia. So now I'll be talking about the layout of our ancient DNA lab. So in ancient DNA lab we have three modules, and we have divided our lab into three parts. An entire lab is positively pressured just to avoid the contamination. So you can see in first room we have a storage as well as the sample processing room, where we expose our archaeological material with the UV light just to avoid the contamination. In second room we have DNA preparation room, and here we are extracting DNA from the archaeological materials as well as the sediment samples. And in third room we have the third room is the most clean room, and here we are processing our DNA libraries and we are amplifying our DNA for the further sequencing. An entire lab is positively pressured and we have pressure gradient. First room we have 20 Pascal pressure, second room we have 30 Pascal pressure, and in the last room we have around 40 Pascal pressure just to avoid a contamination. And here's the real picture of our lab. Here you can see this is the chain room change as well as the storage room. Second room is the DNA extraction room, where here we are processing all the samples. We have one fume hood where we drill all skeletal materials, and all the acid treatment takes place in this fume hood. And this is the most clean part of our lab, where we have two biosafety cabinets, 100% exhaust. Here we are preparing our libraries from the precious archaeological materials. So why we are doing ancient DNA research, and what ancient DNA can tell? So in first objective, we are looking into the population history of a plant, animal as well as human who have lived in the past. And we can also reconstruct the diet pattern of human as well as animal, and what an extinct animal must have eaten over the course of his lifespan, and how extinct plants, animals and humans were related to each other, and what animal were hunted by ancient humans. That information also we can get out of ancient DNA research, and what type of diseases were ancient human population had by analyzing the tooth plaque samples as well as the gut microbiome from the sediment samples. And we can also establish the genetic affinities with the living population and their ancestors. So from any biological specimens we can extract ancient DNA, and it has lot of implications in anthropology and archaeology, and we can also extract DNA from the environmental material, for example soil, sediments, and etc. And these are the main source of the ancient DNA, and we generally prefer the bone samples. If bone samples are not available, we can go for the hair samples. We can also use poo samples, the animal excreta. From there also we can isolate ancient DNA, and from tooth we can isolate ancient DNA as well as the microbiome. So a tooth in past several microbes infects the human individual, and that infection can be found in the form of a signature in the enamel of a person. And we can also extract DNA from the sediments just to see the type of deposition, type of flora and fauna from an ecological niche. So with the recent advancement, we have noticed, and even researchers all over the world, they have found that there is a bone in our skull, in our cranium, that is called petrous bone. So petrous bone has lot of DNA depositions. So nowadays people are preferably using the petrous bone for the ancient DNA extraction, and the second most preferable material is the tooth sample. Even we can extract DNA from the sediment samples. I will give one example from a Ladakh region where we have taken one strata of a sediment where we have found lot of charcoal deposition, and we have sequenced the yolk DNA from this region. And when we have dated this strata, we have found that this strata belongs to 10,000 to 11,000 years before present. So the yolk must have been lived in this strata, and from their excreta we have extracted the yolk DNA, and we have reconstructed the yolk population history in this tough terrain of the Ladakh. So here you can see the signatures of the ancient DNA of yolk. You can see clearly, you can see the base pair damage at the 5' and 3' end of the DNA molecules. So this is a proxy to authenticate our ancient DNA signatures. So now since we have a lot of application of ancient DNA, now I'll give few examples of the human population history what we are working at BSIP. So using the ancient DNA method, we can reconstruct the human population history as well as the demographic expansion of the human populations, and we can also see how human population expanded in a continent. So we are focusing mostly South Asia, and we have sequenced human population remains which has been found from different habitational region of the northwest India and south of India. So I'll give two examples here. And so this is the current scenario of the modern India. You can see we have more than 4,500 well-defined population, which includes 532 tribes, 72 primitive tribes, and we have a lot of diversity in languages, social structure, dress, food habit. So how this kind of diversity have accumulated over time? So we are trying to see the source population of this diversity, and if you want to see the source population, you have to go into past, and you have to look into the past population history. So we have carefully selected few archaeological sites in the northwest of India and South India. I'll give one example of the Indus Valley civilization. So here you can see in the Gujarat and Haryana state, we have lot of prehistoric cities. So these are the map, and here you can see lot of human archaeological sites have been reported over time. We have got lot of archaeologically relevant human remains from a city, old city, that is in Haryana state, and the site name is Rakhigadi. So from this site we have collected 35 human remains. So here you can see they are the burials from the archaeological site of Rakhigadi. From this burial site we have got lot of human remains, and we have extracted the DNA out of the human remains. So for authentic ancient DNA research, you have to have authentic skeletal materials. So to get the authentic skeletal materials should not be contaminated from the modern humans. So we take care of utmost precaution, and here you can see we use aprons and body suits, masks to collect the archaeological materials just to avoid any kind of contamination. So this is a real picture where we have collected the human remains from a Rakhigarhi site. This site is 5,500 year old, and it has yielded around 35 complete human remains, and we are processing these human remains in our laboratory, and we have successfully extracted DNA from these burials. Now I'll be giving another example of a famous civilization in South India. So this is the contemporary of Indus Valley civilization. At the same time period, there were another civilization which emerged during 4000 to 5000 BC, and we call megalithic culture. And this culture has a very peculiar features in terms of the sarcophagus. So they used to bury their dead in a sarcophagus kind of structure, which was made by the stones. And these are the map of this civilization. So you can see mostly this kind of civilization was concentrated in the South India, and we have successfully extracted DNA from 50 burials of these regions, mostly from Tamil Nadu, Andhra Pradesh, Karnataka and Telangana state. And now we are trying to sequence their complete genome, and we are comparing with the modern population just to see the demographic expansion and different kind of selections and natural adaptation of this population. Thank you for watching me, and I hope you have liked my talk.